Hi, my name is Gabriella Schroeder, and today I'm going to be talking to you about the effects of pH on enzyme activity. So a few things I want to go over before we begin is that what is an enzyme? So an enzyme is a protein, and they are produced by cells to act as a catalyst. And when I say that an enzyme is working or acting as a catalyst, it's working to regulate the speed at which a reaction occurs. And to do so, an enzyme will lower the energy required to initiate a reaction, and this is called the activation energy. And doing so allows the rate of the reaction to be carried out faster or sooner. And this is illustrated in a graph right up here. This graph pictured here might help you visualize the reaction. Notice the line in red illustrates the rate of the reaction occurring without an enzyme. Notice how much more energy it takes for the reaction to initiate versus the blue line, which has an enzyme. Notice these peaks are much higher in the red line, which represents it without an enzyme, and how the peak is much lower in the line, which is blue, which represents an enzyme present. Overall, the reaction should occur much faster as it takes less time to build up to the higher activation energy as long as the enzyme is present, as illustrated by the blue line. So another important set of background information to know is what is trypsin, and trypsin is the enzyme we're focusing on in this lab. And trypsin is a protease that is found in the small intestine of humans and animals. We're interested in trypsin because it's believed that trypsin may correlate to neural health, digestive issues, and other diseases. What does trypsin do? Trypsin is important because it aids in the digestion of dietary proteins. It works by hydrolyzing peptide bonds of large peptide chains into smaller peptide chains. This is done by cleaving peptides after the arginine and leucine sequences. Where is trypsin found? Trypsin can be found as it's secreted by bacteria. It is also found in the small intestine of animals, and common conditions that it can be found in includes being active at a pH of 8 in the small intestine, as well as a temperature of 37 degrees Celsius. Moving on to our study. Our question was, how does the pH in a given environment affect the enzyme activity of trypsin? We hypothesized that the enzyme activity of trypsin would be most optimum at a pH of 9. Our rationale for this was in a real-life situation, enzyme activity of trypsin usually occurs in the small intestine at a pH of 8. Considering that a pH of 8 is basic, we thought that enzyme activity would be even more optimum at a more basic pH. Our materials and methods included 400 microliters of reaction buffer, 500 microliters of azocasein substrate, 50 microliters of trypsin enzyme. We then incubated it at 37 degrees Celsius for 20 minutes. We added 500 microliters of 10% trichloric acetic acid, and precipitate was centrifuged at 12,000 rotations per minute for 2 minutes. A spectrophotometer was then used to measure the absorbance of the supernatant at 440 nanometers. Important notes to know about our materials and methods. The substrate, azocasein, is a milk protein. It absorbs light best at 440 nanometers because of the sulfonylamide groups it contains. We added TCA to stop the reaction and precipitate undigested substrate. We centrifused the reaction after adding the TCA to remove the undigested substrate. The sulfonylamide was added to the precipitate because it allows the molecules to absorb the light at 440 nanometers when we were using the spectrophotometer. Continuing with our methods, the experimental variable was the pH of the solution. We tested this at pH values 8, 9, and 10. Our control variables included a salt concentration of 0.5 moles of sodium chloride and a temperature of 37 degrees Celsius. Our negative controls were used to show no enzyme activity. We added no trypsin to negative controls. Loss of volume was made up with an additional 50 microliters of reaction buffer. This graph illustrates the absorbance values of trypsin at the 440 nanometers. Two important observations can be made with this graph. The first being that the control values, found in orange, show little to no absorbance as there is no enzymes found there. The second being that there is a lot of variability in the absorbance values, meaning that the results do not definitively show a correlation. Here pictured above should be our t-test results. Our t-test results are important in proving if a pH of 9 is optimum. If they don't show a correlation to enzyme activity, then we can't prove our hypothesis. So pictured here should also show our experimental groups versus our negative control. This is important in establishing that yes, without the enzyme present, there should be no activity. And our results did prove this, as the p-value was less than 0.05, so the amount of enzyme activity in the experimental group is significantly higher than the control group, meaning that yes, enzyme activity does happen when an enzyme is present. This is also proven at pH 8, 9, and 10 with the negative controls. And then we tested pH 9 experimental versus the pH 8 experimental, and the p-value was a lot greater than 0.5. Here you can then evaluate our experimental values. At pH 9 experimental versus pH 8 experimental, you can see that the enzyme activity is not significantly greater because the p-value is greater than 0.05, so there is no correlation. The same thing happens with a pH 9 versus a pH 10 experimental, where the p-value was 0.063, so again that's much greater than the 0.05, again proving that a higher pH is not optimum for trypsin. So moving on to our conclusion, as I just stated, we originally hypothesized that our pH 9 would be most optimum, and at first we thought our hypothesis was correct. However, after conducting our t-test result, we discovered that enzyme activity was statistically significant between the experimental and control groups, but not between comparing pH 8, 9, and 10. So there was no significance, therefore proving our hypothesis wrong. So moving on from the conclusion and into the discussion, I'm going to talk about how we can improve our experiment and how we did that from the first stage to the second stage. As I said, we felt like our experiment was more adequate in the second round. The first round, we really based it off of how we had 5, 6, 7, 8, 9, 10 pH levels that we were testing, and we were only testing one replicate of those, each with a control group. And so there were several changes that we made to that. First of all, we ended up conducting in our second and final round of experimenting only at a pH of 7, 8, and 9. And the reason we were able to establish this was from data from other groups. We were able to obviously eliminate 5 through 7 as well as 10 because we really didn't see any enzyme activity at those levels, and we want to express where it's most optimum. So that's not going to really help us prove that it's most optimum if there's not a lot of activity. We then moved on to adding the 3 replicates, and the importance of adding those 3 replicates for each trial was to reduce variability and then also have a more accurate, average, and precise measurement. And this is important in proving the hypothesis and really understanding like a cause, because if you only have 1 replicate for each experiment, you really can't see if there is errors or even like variance and maybe something was done wrong here. And so one thing we then learned how we could improve this experiment was based off of the results of other groups, that we were able to determine that another way we can improve this experiment was testing at a pH of closer to 47 degrees. That one group found that tested that the 37 degrees Celsius is not the most optimum, and there was higher activity at 47 degrees. And we would also use more replicates. Again, this is important in reducing that variability and understanding if there is a correlation. Again, to close off with this, the results of trial 2 were slightly higher than those of trial 1 and 3. So our values for average and variability were both higher as a result of that. Since our data showed that the pH 9 did not show significantly higher enzyme activity, we can conclude that the ideal condition to grow bacteria would not be as basic as we previously thought, and it would not be optimum at a pH of 9.